Hi friends, welcome back to my channel Only Bible, presenting up the second part of revision of the chapter Excretion and Osmoregulation. A very important chapter. So friends, in this particular series of excretion and osmoregulation as per the change syllabus, I am going to divide it into many parts. So please keep a check on the playlist and keep following this series for better understanding. Your queries, your suggestions are most welcome. So, if you remember in the last lecture, we had a talk about the modes of excretion, and we also said that the modes of excretion are largely influenced by the habitat because this is nothing but the availability of the water for that organism. So, the first mode of excretion is ammonotelism. The elimination, that is removal of nitrogenous waste in the form of ammonia, is called as ammonotelism. Ammonia is formed directly in the process of catabolism of amino acids and it is basic in nature. So, if ammonia gets accumulated, it can disturb the pH of the body. Okay. Slight increase in the pH would disturb all enzyme catalyzed reactions in the body and would also make the plasma membrane unstable. Friends, let's understand what exactly this statement means. Even though there is a slight increase in the pH, you know that we all say that the body needs to maintain a homeostatic state, that is a balanced state. A little higher or a little lesser than the optimum can cause a problem in the body. So, pH is one of such factors which can disturb the homeostatic balance. If the pH slightly increases, it is quite possible that the enzymes get deactivated. You know, these all are the enzymes that control each and every chemical reaction that do take place in our body. So certain enzymes require acidic pH, some enzymes require a basic pH, while some require a neutral pH. So all those enzymes are likely to be disturbed if at all nitrogenous waste products are retained in the body, they accumulate in the body, and this can disturb the pH. And because of this, the plasma membrane becomes unstable. Now, friends, if the plasma membrane becomes unstable, there is a possibility that the substances that are unwanted might enter the cell, and the substances which are crucial, which are important for the cell, might exit the cell, right? And this can further cause a problem within the cells. Ammonia is readily soluble in water and needs large quantity of water to dilute and reduce the toxicity. However, it is energy saving mechanism of excretion. Now, why do we say this? It is readily soluble in water, no doubt, but it requires large quantity of water. So here water consumption is the highest. Now, why large quantity of water is essential? So that the ammonia can be converted to what you can say a diluted form wherein the ammonia concentration might not really prove to be toxic. Okay, now why is it said to be energy saving mechanism? The reason is ammonia being readily soluble in water, it easily diffuses out. So the body does not spend more energy to throw it outside the body. Hence, all animals that have plenty of water available for dilution of ammonia excrete nitrogenous wastes in the form of ammonia, and such animals are called as ammonotelic animals. 1 gram of ammonia needs about 300 to 500 ml of water for elimination. From this, you can get an idea that it's a big volume of water that is going to be utilized for the elimination. Ammonotelism is found in aquatic invertebrates, bony fishes, and aquatic or larval amphibians, aquatic insects. Now, friends, if you did observe, I had stressed on the word bony fishes. The reason behind it is cartilaginous fishes do not show ammonotelism, but they show ureotelism. No doubt, कि fishes are aquatic, but again, there is a category. It could be either marine water fishes or it could be freshwater fishes. And exactly that can influence the mode of excretion. Animals without excretory system are also ammonotelic. For example, protozoa. Ammonotelic animals excrete ammonia through general body surface, that is skin, gills or kidneys. Friends, you should be remembering here it easily diffuses out. So it can easily escape via the body surface, via the skin, the gills. But kidneys do not really play a major role here. Okay, kidneys have a minimum role to play in this process. Ammonia being readily soluble, it is generally excreted by diffusion across the body surfaces or through the gill surfaces in the fish as ammonium ions. Kidneys do not play a significant role in its removal. Now the next is ureotelism. Elimination of nitrogenous waste in the form of urea, that is H2NCONH2, or rather CONH2NH2. You might have done this in your chemistry. This is called as ureotelism. Urea is less toxic. Matlab, it is less toxic as compared to ammonia and less water soluble than ammonia. Hence, it can be concentrated to some extent in the body. Friends, try to understand ammonia being highly toxic, it has to be thrown out as soon as it is formed. But urea being less toxic, it can be stored in the body to some extent. Due to this, it requires less water for elimination compared to ammonia, about 100 times less water in human, several hundred times less in camel, kangaroo rat and shark. Now friends, on the screen you can see a kangaroo rat. There are many students who really wrongly interpret this particular sentence, and while they read and while they write, they put up a comma in between kangaroo and rat and they read it as kangaroo and rat. But it isn't so. This is a variety of rats. Okay, and they're called so because their mode of movement and locomotion, the way they run, is very very similar to the hopping movement of that of the kangaroos. So these rats are called as kangaroo rats, and they have a special feature that they can survive without water for quite a long time. Now you may feel, how is that possible that an organism can survive for a long time without water? The reason is this kangaroo rats, they feed on seeds, and the seeds or any plant material that they feed on contains ample quantity of water which is sufficient enough for the metabolic reactions to run. As it is less toxic and less water soluble, and hence ureotelism is suitable for animals, those need conservation of water to some extent. In other words, you can say that it is an adaptation for the survival on the land. Hence, ureotelism is common in terrestrial animals as they need to conserve water. It takes about 50 ml of water for removal of 1 gram of ammonia in the form of urea. Mammals, cartilaginous fishes like sharks and rays, many aquatic reptiles, most of the adult amphibians, etc., are ureotelic. They convert ammonia to urea in the liver by operating ornithine, that is urea cycle, or Krebs and Hensley cycle. Now friends, please remember nitrogenous compounds can be stored in the body. So the excess of the nitrogenous compounds are first carried to the liver where they are going to undergo a process called as deamination. Now DED is removal, amination is amino group. So the amino group, that is NH2 group, is separated from these nitrogenous compounds in the liver by a process called as deamination. Okay, and then this amino group may react with hydrogen to form ammonia, or it may react with carbon dioxide to form urea, and that process is called as the ornithine or urea cycle, which was discovered by Krebs and Hensley in 1932. 3 ATP molecules are used to produce 1 molecule of urea. A very very important point in terms of competitive exams, ये MCQs में आ सकता है. Sharks retain more urea in their body fluid, that is blood, to make their blood isotonic to the surrounding marine water, and this helps them to prevent possible loss of water by exoosmosis. Now friends, sharks are marine water fishes, right? So marine water, that is salty water. So अगर sea water की बात हम करें तो what do we find here is the concentration of solutes is quite higher as compared to the concentration of solvents. Now this I'm talking in comparison to shark. So shark, imagine the shark to be in the marine water. Obviously it is its natural habitat, but sharks in comparison to the marine water is ought to have less salts and more water. So what is the possibility? You know, water has a tendency to flow from higher to the lower level. So in this case, it is possible that the excess of the water from the body of the shark may diffuse out. That is, it can undergo exosmosis. Now, if this water escapes out from the body, it is likely to shrink and die. But does that happen? No. The reason is sharks retain urea in the blood. So, what happens is urea, which is a solute, when it is retained in the blood, but obvious, the concentration of solute goes on increasing till it becomes isotonic, that is, having the same balance with that of the surrounding water. So this is how it maintains its homeostatic balance. In the liver of ureotelic animals, ammonia is combined with carbon dioxide to form urea. The reaction is 2NH3 plus CO2 gives you CONH2 twice plus H2O. Urea is 1 lakh times 1 lakh times less toxic than ammonia. Please remember this. Urea is 1 lakh times less toxic than ammonia. This conversion requires expenditure of energy. That is conversion of ammonia into urea. Even then these animals can afford it because urea can be stored for long and can be excreted at a slower rate with the availability of water. Urea is carried by the circulatory system from the liver to the kidney for the elimination. However, in mammals, all the urea produced is not excreted immediately. Some portion of it is retained in the kidneys for osmoregulation. That's why I have taken up the example of shark. I hope you have understood it. These are mainly semi-aquatic or terrestrial animals like frogs, toads, and mammals, respectively. Ammonia produced by metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidneys. From this, you have to understand that it is formed in the liver, but it is collected and thrown out by the kidneys. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity, that is, concentration. Now, the next one is uricotelism. It is the elimination of nitrogenous waste in the form of uric acid, that is C5H4N4O3. I repeat, C5H4N4O3. Synthesis of uric acid from ammonia requires far higher expenditure of energy than the synthesis of urea. Okay, so here energy consumption is more, but water consumption is going to be lesser. It is advantageous to land animals as it is far less toxic and hence can be retained in the body for some time in concentrated form and insoluble in water. Minimum 5 to 10 ml of water per gram or no need of water at all. Hence, it does not require much water for its elimination and can be excreted in the form of a paste. It may be paste or a pellet-like thing, a powdered crystalline thing. Organisms excreting uric acid are called as uricotelic animals, and these are terrestrial animals like the land snails, the land reptiles, the birds, etc. Birds का तो काफी बार आपने शायद कभी तो जिंदगी में एक बार एक्सपीरियंस किया ही होगा. Uricotelism is particularly advantageous to the oviparous land vertebrates like birds and animals that need to conserve more water. As I said, it can be considered to be as a kind of an adaptation to the existing habitat. Birds lay shelled eggs on the land, that is, their eggs have a shell. Ammonia and urea can pass out of shellless eggs of Pisces and amphibia which are in water. Okay, they do not have shells, so that can easily pass out. Egg shell of birds has pores only for gaseous exchange. Had an embryo produced ammonia or urea within the shelled egg, the soluble waste would have accumulated to the level of toxic concentration. Try to understand. Okay, so that would have killed the embryo. But instead, uric acid is going to be produced in these. So, uric acid precipitates out of the solution and can be stored in the shell as a solid waste. It is left behind when the young one hatches out. Ammonia is converted into uric acid by inosinic acid pathway in the liver of the birds. जैसे ureotelism, what did we see was Krebs lead cycle. Here we find inosinic acid pathway. Birds, some insects, many reptiles, land snails are uricotelic because they need to conserve the water. However, they need to spend more energy. They excrete the nitrogenous waste as uric acid in the form of pellets or paste with a minimum loss of water. Inosinic acid metabolic pathway is responsible for the synthesis of uric acid from ammonia in these animals. And this pathway was first discovered by Buchan and his co-workers in the 1950s in pigeon liver. Very, very important. Buchanan. Many insects store excretory products in their body instead of eliminating them. This phenomena is called as storage excretion. Friends, this point is nowhere there in our textbook, so please, it's advisable to note it down. It saves expenditure of water during excretion. Since uric acid is non-toxic and highly insoluble, it can be retained in the body for a long time without any ill effect. Cockroaches are adapted to this technique and they store uric acid in the fat body and the cuticle. I've deliberately taken this point because you also have a chapter on cockroaches, right? So this point can be related there and you can understand it better. In turn, the stored uric acid provides nitrogen deposits for the mobilization at the time of nitrogen deficiency. Friends, there's another mode of excretion which is little heard and that's guanotelism. Animals like spiders, scorpions, and penguins excrete guanine. So, this mode of excretion is called as guanotelism. Guanine is a nitrogenous base. The formula is C5H5N5O. Like uric acid, guanine is a purine, relatively non-toxic and insoluble in water. Thus, it is also excreted in the solid form. Friends, if you cross-check in few books, they have written that uricotelism itself is called as guanotelism, but it isn't so. Uric acid and guanine, okay, no doubt both are purines, but their chemical formula is slightly different. So, ideally, uricotelism and guanotelism are slightly different from each other. It is an adaptation to life in the arid habitats, that is, dry habitat without moisture. In guanotelic animals, the inosinic acid pathway terminates in guanine formation. So, from this, you can understand that it's the inosinic acid pathway itself which is going to operate there. The term guanine is derived from the Spanish word guano, which means bird or bat droppings. Guanine is considered to be rich in nitrogen. The white fecal matter in the guano consists of uric acid and urate crystals which are insoluble. Hence, guano is used commercially to produce uric acid. Parts of an island off the coast of South America is covered with guano. You wouldn't imagine, but literally people are appointed to break down these guano deposits so that they can extract uric acid from the same. Now, friends, this is a comparison between ammonotelism, ureotelism, and uricotelism. This would help you to answer up the questions like distinguish between. So, I have just highlighted it here. I shall read it out. You will understand because we have already gone through it. So, first point, the elimination of nitrogenous waste in the form of ammonia is called as ammonotelism. The elimination of nitrogenous waste in the form of urea is called as ureotelism, and the elimination of nitrogenous waste in the form of uric acid is called as uricotelism. Second point, the organisms that excrete ammonia are called as ammonotelic organisms. The organisms that excrete urea are called as ureotelic animals, whereas the organisms that excrete uric acid are called as uricotelic animals. Point number 3. Ammonia is highly toxic and highly soluble in water. Urea is less toxic than ammonia but more toxic than uric acid, and it is slightly soluble in water. Uric acid is the least toxic and insoluble in water. Point number 4. Very little amount of energy is used in the formation of ammonia. More amount of energy is used in the formation of urea, and maximum amount of energy is used in the formation of uric acid. Point number 5. Ammonotelism includes considerable loss of body's water. Ureotelism leads to less loss of body's water, and uricotelism causes least loss of body's water. Friends, this point can also be written as water consumed is high and conserved is less. In that format, you can write it in your own words. Point number 6. Ammonotelism occurs in aquatic animals. Ureotelism occurs in aquatic as well as in land animals. Uricotelism occurs in land animals. Point number 7 are the examples. For ammonotelism, we have amoeba, sypha, hydra, earthworm, unio, prawn, salamander, tadpole of frog, and bony fishes. The examples for ureotelism, we have earthworm, cartilaginous fishes, frog, toad, turtles, alligators, mammals, whales, seals, etc. And the examples for uricotelism, we have insects, land crustaceans, land snails, land reptiles, birds, and snakes. Point number 8. Ammonia is formed in the liver by the deamination process. Urea is formed in the liver by the ornithine cycle, and uric acid is formed in the liver by inosinic acid pathway. Point number 9. Ammonia is excreted rapidly in the urine. Urea is excreted at a comparatively slower rate, and uric acid is excreted very slowly. Point number 10. Ammonia is excreted in the form of dilute urine. Urea is excreted in the form of concentrated urine, and uric acid is excreted in the form of pellets or thick paste. Point number 11. 500 ml of water is required for elimination of 1 gram of ammonia. 50 ml of water is required for the elimination of 1 gram of urea. 5 to 10 ml of water is required for the elimination of 1 gram of uric acid. Friends, I know it was a long one, but hope you have liked it. Please subscribe if you are new to the channel and hit the bell icon. Please share it with your friends. Thank you so much for being with me.